Imagine you're, I don't know, preparing for a biological war,
Okay.
and you decide to put on an extra layer of armor because intuitively, like covering yourself in a thick defensive cloak should make you tougher, right? Harder to kill.
Yeah, you definitely think so.
Right. But in the microscopic world, adding that extra layer of armor actually makes you incredibly fragile.
It really does.
It turns this relentless, indestructible threat into something that can be, well, completely annihilated by a simple bar of soap.
It is the ultimate structural paradox. I mean, the very layer that allows some of our most notorious pathogens to infiltrate our cells is also their greatest vulnerability out in the open environment.
So today, we are tearing down the architecture of viruses.
Yeah.
Welcome to another deep dive.
Today's stack of sources is all about microbiology, specifically zeroing in on virus structure and classification.
It's a huge topic. It really is. We are going to decode how these entities are built from first principles. We'll explore the bizarre century-old debates around whether they even qualify as living things, and look at why mapping their physical geometry is basically the foundational cheat code for modern medicine.
Yeah, because viruses operate under extreme constraints. They are the ultimate minimalists. By stripping away everything but the absolute biological necessities, they reveal these fascinating survival strategies that challenge our very definitions of life.
Okay, let's unpack this, because before anyone could analyze how a virus is built, scientists had to deduce they even existed, right? Through, like, negative space.
Exactly. They couldn't see them.
Right. So in the 1890s, the absolute pinnacle of microbiological tech was this thing called the Chamberlain filter.
Unglazed porcelain filters.
Yeah, with pores so incredibly microscopic, they were guaranteed to trap any known bacterium. So if you passed a diseased liquid through it, the output was supposed to be perfectly safe, sterile water.
That was the gold standard back then. If you suspected a bacterial pathogen, the Chamberlain filter was your ultimate diagnostic tool. The assumption was just that life, even microbial life, had a minimum physical footprint.
But then in 1892, this Russian botanist, Dmitry Abanovsky, is studying tobacco plants that have this mosaic disease.
Right, the tobacco mosaic
Yes.
disease.
He mashes up the infected leaves, runs the sap through this state-of-the-art porcelain filter, and the liquid that comes out the other side, it still aggressively infects healthy plants.
Which completely broke the current understanding.
But his conclusion wasn't that he'd discovered a new biological domain. He just assumed his filter was broken, or that the bacteria were secreting some kind of chemical toxin that slipped through.
Which, to be fair, was a highly logical deduction for the 1890s. The paradigm of the time just couldn't accommodate a particulate organism smaller than those pores.
Right.
It took Martinus Beijerinck in 1898 to really push the experimental boundary. He repeated Ivanovsky's work, but he added this crucial step. He proved the infectious agent could diffuse through a solid agar gel.
And bacteria don't do that.
Exactly. Bacteria don't do that.
Yeah.
Toxins might, but this agent actually multiplied. So Beijerinck realized this was something categorically different from bacteria. He described it as a contagious living fluid.
A fluid.
Yeah, and he popularized the term virus, which is Latin for poison or slimy liquid.
It's basically like trying to catch sand in a pasta colander.
That is a perfect way to look at it.
You expect the colander to trap everything solid, and when the sand just washes right through, you have to completely rethink what solid even means in this context. And right around that same time, Friedrich Loeffler and Paul Frosch demonstrated the same filterable phenomenon with foot and mouth disease in cattle.
So it wasn't just isolated to botany, it was this universal biological threat.
It was everywhere.
But just identifying that something passes through a filter doesn't tell you what it actually is. I mean, this filterable agent lacked metabolism.
Right.
It didn't consume energy, it didn't excrete waste, it didn't grow, and it couldn't reproduce on its own in a petri dish.
Which brings us to 1935, and a moment that essentially broke the definition of biology entirely.
Oh, yeah, Wendell Stanley.
Yeah. Wendell Stanley takes the tobacco mosaic virus and manages to crystallize it. Now, wait, so a virus outside a cell is biochemically closer to a grain of salt than a living germ?
Yes. And Stanley actually won a Nobel Prize for that crystallization because it seemed to definitively prove viruses were just complex proteins, inert chemicals rather than organisms.
Because you can't crystallize a living, metabolizing cell.
Exactly. A crystal is frozen. It has no biological agency. But what's fascinating here is that the paradox, and the reason the debate still rages today, is that once that inert chemical lattice enters a host cell, the resulting cascade is indistinguishable from life.
It wakes up.
Right, it directs an active, evolving, highly complex replication process.
It really forces us to accept this dual-state existence. Many modern microbiologists look at it as two distinct phases, right?
Exactly.
You have the dormant particulate phase, the virion, when it's just floating outside a cell, and then the active viral phase of borrowed life once it actually penetrates the host and hijacks all the replication machinery.
And because it has no machinery of its own, like no ribosomes, no mitochondria, every structural choice the virion makes has to perfectly serve two singular mandates.
Just two jobs.
Just two. Protect the genetic payload at all costs and successfully deliver that payload into the next host.
So let's examine that payload. In cellular life, the genetic storage medium is pretty standard. It's double-stranded DNA. But viruses use this completely wild mix-and-match approach to their genomes.
They really do.
You see single-stranded DNA, double-stranded RNA, linear strings, circular loops, or even segmented genomes broken into separate pieces, almost like a hand of playing cards.
They exploit every possible nucleic acid combination. I mean, no cellular organism uses single-stranded DNA or double-stranded RNA as its primary genetic repository.
Yeah.
But viruses do it routinely.
Wow.
And the size constraints vary wildly based on their evolutionary strategy.
Yeah. Right. The sources highlight the porcine circovirus, which operates on this incredibly tight budget of about 1,700 nucleotides.
Tiny.
Yeah, tiny. But then on the absolute opposite end of the spectrum, you have the Pandora viruses, discovered around 2013, and they are packing over 2.5 million base pairs.
It's massive.
That is a larger genetic footprint than many free-living bacteria.
It is. But regardless of whether it's an incredibly compact 1,700 nucleotides or a sprawling 2 million base pairs, that fragile nucleic acid has to be shielded from environmental degradation, UV radiation, host enzymes.
So it needs a box.
Exactly. This necessitates the second structural layer, the capsid.
The capsid is this protective protein shell. But if a virus has a tiny genome, it obviously can't afford to encode instructions for a massive, custom-shaped, contiguous shield. It has to utilize geometric efficiency.
It relies heavily on self-assembling repeating units.
I look at it like flat-pack furniture engineering.
Okay, I like that.
Like, if you order a massive bookshelf, the manufacturer doesn't ship you a pre-built hollow wooden box. That would be completely inefficient to transport.
Right, you get the flat box.
Exactly. They ship a dense, flat box containing dozens of identical wooden panels and pegs that are just designed to snap together. The virus genome operates the exact same way. It doesn't encode a single giant shell. It encodes a few tiny identical protein subunits called capsimeres that just automatically fold and snap together into a symmetrical enclosure.
The flat pack analogy perfectly illustrates the constraints of viral genetics, because the geometry of self-assembly dictates that capsids can only form into a few mathematically stable shapes.
They can't just be anything.
No. In 1962, Donald Caspar and Aaron Klug formalized this with their quasi-equivalence theory. They basically demonstrated the strict mathematical logic behind why these capsids are built from repeating triangular facets, forming these predictable geometric tiers.
So they don't just arbitrarily increase in size. They have to jump from one specific mathematical volume to the next, almost like upgrading a geodesic dome by adding a specific mathematical ratio of new triangles.
Precisely. And that underlying geometry results in three primary capsid architectures.
Okay.
You have the helical shape, where capsimeres form a rigid tube or a flexible coil spiraling around the genome. Rabies and the tobacco mosaic virus use this.
Right.
Then you have the icosahedral shape, which is a 20-sided sphere. Mathematically, an icosahedron is the most efficient way to enclose a maximum internal volume with the smallest number of repeated flat parts.
Efficiency is key.
Yep. Poliovirus and herpes viruses utilize this structure.
And then the third category is the complex capsid, which just looks entirely alien to me.
Oh, the bacteriophages.
Yes, the bacteriophages that target bacteria. They resemble lunar landers. They feature this icosahedral head containing the DNA, sitting on top of a helical tail sheath that functions like a hypodermic needle.
Complete with spider-like tail fibers to physically dock onto the bacterial wall.
It's wild.
It is. But what's really remarkable is that these precise architectures remain completely theoretical for decades. It wasn't until Rosalind Franklin applied X-ray crystallography to viruses in the 1950s that we could actually confirm these shapes.
Right, because you can't just put a virion under a standard light microscope. You have to take those viral crystals Wendell Stanley made, shoot a beam of X-rays through the static molecular lattice, and capture the diffraction pattern. Basically, the shadow the atoms cast as the X-rays bounce off them.
And Franklin's diffraction images of the tobacco mosaic virus proved it was a hollow helical tube. Combined with the commercialization of the electron microscope, we finally moved from visualizing an invisible infectious fluid to mapping a precise geometric blueprint.
Which brings us back to that armor paradox we started with.
Yeah.
We have the genome and we have the flat-packed protein capsid protecting it. These are naked viruses, but some viruses deploy a third layer. And they don't even synthesize this layer themselves. They steal it from their host upon exit.
The viral envelope. As an enveloped virus buds out of the host cell, it pushes against the host's lipid bilayer membrane. It basically wraps itself in a piece of that host membrane, severing it to form a stolen cloak.
A stolen cloak of fat.
Exactly. It then embeds this lipid envelope with its own viral glycoproteins.
Like the spike protein on SARS-CoV-2.
Right, or the hemagglutinin on influenza. These glycoproteins act as the keys to unlock the next host cell.
So structurally, HIV, influenza, COVID-19, and herpes are enveloped viruses. They have a genome, a capsid, and this stolen lipid membrane. But poliovirus and norovirus are naked viruses, meaning they stop at the protein capsid.
Yes.
But here's where it gets really interesting. My initial intuition, and I assume a lot of people's intuition, is that the enveloped virus with all those layers is the more heavily fortified, dangerous entity out in the environment.
The lipid paradox dictates the exact opposite. That envelope is composed of host cell lipids, fat molecules. Out in the physiological environment of a host body, it functions perfectly. But exposed to the external environment, a lipid bilayer is incredibly unstable.
Because any basic detergent, mild soap, or standard alcohol-based hand sanitizer immediately dissolves fat.
Yes, the alcohol shreds the lipid membrane. And because the virus's critical glycoproteins, its targeting and entry systems are physically anchored in that envelope, dissolving the fat washes away the keys.
Oh, wow.
The underlying capsid might still be totally intact, but without the glycoproteins, the virion cannot attach to a new cell. It becomes biologically inert.
Which is why enveloped viruses typically require close contact, respiratory droplets, or fluid exchange to spread. They cannot survive for long periods on a dry, hostile surface. But the naked viruses, lacking that fragile layer of fat, are essentially microscopic tanks.
That's right. A naked capsid is just a tightly interlocked protein shell. Alcohol does not readily dissolve those protein bonds, so they withstand harsh environmental conditions, surviving on dry surfaces and water systems, and resisting mild disinfectants for extended periods.
So wait, when millions of dollars are spent distributing alcohol-based hand sanitizers during a norovirus outbreak on a cruise ship, we are essentially just giving the virus a bath.
Pretty much.
We're fighting the wrong architecture. Norovirus is a naked virus. The alcohol won't breach the capsid.
The structural reality of norovirus completely invalidates the alcohol sanitizer response. To neutralize a naked virus on your hands, you have to rely on the mechanical friction of vigorous hand washing with soap and water to physically flush the virions down the drain.
Or use aggressive bleach-based disinfectants on surfaces to chemically denature the capsid proteins.
Exactly.
So if a single microscopic layer of stolen fat completely alters the survival capacity and transmission vector of a pathogen, it makes total sense that the medical community had to rethink how we categorize these threats. I mean, for centuries, we categorized them by the symptoms they cause, right? Hepatitis viruses affecting the liver, respiratory viruses causing colds.
And symptom-based taxonomy is clinically useful for initial diagnosis, sure, but it fails to inform treatment or containment. I mean, two viruses can cause severe respiratory distress, but possess entirely different structural vulnerabilities.
So taxonomy shifted strictly to structure and mechanism. The International Committee on Taxonomy of Viruses, the ICTV, established a Linnaean hierarchy, sorting them by realm, kingdom, and family based on their physical builds. But the system that truly maps their operational mechanics is the Baltimore classification, introduced by David Baltimore in 1971.
Yeah, the Baltimore system abandons symptomology entirely. It categorizes all viruses into 7 classes based on a single universal biological hurdle: the pathway the viral genome must navigate to generate messenger RNA.
Because regardless of whether a virus packs single-stranded DNA, double-stranded RNA, or anything in between, the host cell's ribosomes only read one language.
Guy.
They only translate mRNA. So if the virus can't convert its specific genetic payload into mRNA, the host cell just won't manufacture the viral proteins.
It acts as a mechanical choke point. The Baltimore class predicts exactly what enzymatic tools a virus must carry with it to clear that hurdle.
It's like categorizing vehicles by their internal combustion mechanism rather than their paint color.
That's a great way to put it.
If I know a vehicle requires diesel instead of unleaded gasoline, I instantly know what type of fuel pump it needs, how the engine block is reinforced, and basically how to disable it.
Take a class 5 virus like Ebola or the rabies virus. Their genome consists of negative-sense single-stranded RNA. Now, negative-sense RNA is essentially the mirror image of mRNA. A host ribosome cannot attach to it or read it,
It's backwards.
basically. Furthermore, the human host cell contains no internal enzymes capable of transcribing negative-sense RNA into positive-sense mRNA.
So a class 5 virus is fundamentally incompatible with the host's default machinery. It has to bring its own mechanic.
Yes. The virion must physically package a custom enzyme, an RNA-dependent RNA polymerase, inside its capsid alongside the genome. When it breaches the cell, it uses its own polymerase to flip the negative-sense RNA into readable mRNA.
Wow.
If a mutation prevents the virus from packing that specific enzyme, the genome remains permanently unreadable.
And by isolating these strict mechanical rules, we also expose the biological anomalies that break them entirely. We mentioned the Pandora viruses earlier, but the giant viruses, like mimivirus discovered in 1992, they completely blur the line between viral and cellular life.
They do. Mimiviruses are so physically massive they were initially misclassified as bacteria.
Really?
Yeah. But more disruptively, their massive genomes encode hundreds of genes, including components for protein translation.
Wait, I thought viruses didn't have translation machinery.
Historically, the absolute defining line of a virus was its lack of translation machinery. But giant viruses possess remnants of this machinery. This sparked intense debate among researchers like Jean-Michel Claverie and Didier Raoult regarding whether they represent a defunct fourth domain of life that devolved from ancient cellular ancestors.
The boundaries get even stranger when you look at the extreme edge cases, viroids and prions.
Oh, yes.
Theodore Diener discovered viroids in 1971. A viroid dispenses with the capsid completely. It is merely a naked circular loop of RNA.
Just RNA.
No protein shell, no structural geometry, just a raw snippet of infectious code that somehow manages to devastate agricultural crops, like causing potato spindle tuber disease.
And if we connect this to the bigger picture, prions sit on the absolute inverse side of that spectrum. Identified by Stanley Prusiner, prions dispense with the genome entirely. They possess zero genetic material, no DNA, no RNA. They are solely composed of misfolded proteins.
So if a standard virus is a rogue instruction manual that infiltrates a factory to print more manuals, a prion is just a malicious typo in the factory's existing machinery.
Exactly.
It's a contorted protein that physically bumps into normal, healthy proteins and just forces them to adopt its misfolded shape.
And that cascading chain reaction of misfolding leads to fatal neurodegenerative conditions like scrapie in sheep or Creutzfeldt-Jakob disease in humans. Prions and viroids are crucial to microbiological theory because they isolate the variables. They demonstrate the extreme endpoints of parasitic replication.
Proving that the standard genome plus capsid model of a virus is just our most common structural observation, not an absolute boundary of nature.
Well said.
So what does this all mean for you listening? Understanding whether a pathogen is a capsid-protected RNA strand, a lipid-wrapped DNA genome, or a rogue misfolded protein isn't just an exercise in academic sorting. This structural knowledge forms the literal engineering foundation for modern medical intervention.
It separates broad, scattershot treatments from precision molecular targeting. Consider the persistent misuse of antibiotics. Patients frequently request antibiotics for viral infections like the common cold.
But antibiotics are chemical weapons designed to target the structural anatomy of bacteria. They target the bacterial cell wall or jam the specific mechanics of bacterial ribosomes.
Exactly.
And as we've thoroughly established today, a virus doesn't construct a cell wall and it doesn't possess ribosomes. Throwing antibiotics at a viral capsid is structurally meaningless.
Right. To neutralize a virus therapeutically, you have to design an antiviral drug that chemically binds to and jams the highly specific viral enzymes dictated by its Baltimore classification.
And the mapping of HIV in the early 1980s by researchers like Luc Montagnier and Robert Gallo is the ultimate testament to this, right? By mapping its structure and genome, they identified HIV as a Baltimore class 6 virus, a retrovirus.
And the defining characteristic of a retrovirus is that its genome is RNA, but it carries a unique enzyme called reverse transcriptase. Upon entering the cell, it uses this enzyme to copy its RNA backward into double-stranded DNA.
Which is wild.
It reverses the standard flow of cellular biology, which typically moves from DNA to RNA to protein.
But because they identified that exact structural mechanism, an enzyme that doesn't exist in normal human cells, pharmacologists knew exactly where to strike. They engineered antiretroviral drugs, specifically reverse transcriptase inhibitors, that slot right into that viral enzyme and break it, halting the replication process without damaging the host's normal functions.
However, that same structural mechanism explains why HIV remains so difficult to cure. Once a reverse transcriptase converts the viral RNA into DNA, another viral enzyme integrates that new DNA directly into the host cell's own chromosomes.
But the viral code weaves itself seamlessly into the human blueprint.
Exactly.
You can't just wait for the immune system to flush it out like influenza because the virus has become a latent physical part of the host's genetic architecture.
And our entire framework of pandemic preparedness relies on mapping these precise architectures. The COVID-19 mRNA vaccines were developed with unprecedented speed, not because of guesswork, but because within weeks of sequencing the SARS-CoV-2 genome in early 2020, structural biologists mapped the exact atomic geometry of the spike glycoprotein embedded in its lipid envelope.
The vaccine simply delivered the mRNA instructions to synthesize that specific 3D shape, training the immune system to recognize the architecture before the actual virus ever arrived.
Yep.
It is a war fought entirely through structural intelligence. I mean, from Ivanovsky's realization that the Chamberlain filters were letting an entirely new class of pathogens slip through, to Wendell Stanley crystallizing the invisible, all the way to understanding why a stolen lipid envelope makes a deadly pathogen vulnerable to hand sanitizer, while a naked capsid remains completely unfazed.
And, we've discussed how retroviruses like HIV integrate their genetic code directly into the host chromosome, becoming a permanent fixture of that specific cell. If you scale that mechanism across deep evolutionary time, the implications are staggering. Retroviruses have been infecting living organisms for hundreds of millions of years. Occasionally, a retrovirus infects a germline cell, like a sperm or an egg.
Meaning that integrated viral DNA gets passed down to the offspring.
Exactly. It becomes a permanent genetic inheritance, generation after generation, species after species, accumulating these deactivated latent viral insertions. When geneticists sequence the modern human genome today, they find that approximately 8% of our entire DNA is composed of ancient endogenous retroviruses.
8%. That is crazy. We spend all our time mapping virus structures to fight them off, completely unaware that a massive chunk of our own biological blueprint is built from the ancient fossilized code of the very entities we are fighting. We aren't just battling viruses, we are partially constructed by them.
It really makes you think.
A perfect, slightly unsettling reality to leave you with today. Thank you for joining us on this deep dive into the architecture of the invisible world. And for you listening, definitely remember the difference between naked and enveloped structures the next time you decide between hand sanitizer and a sink full of soap on a cruise ship. We'll catch you next time.